So welcome technicians. Today we look into one of the main biochemistry instrument in our Indiana lab, that is the COBA C311 instrument. So this is the instrument. So again, this is also an instrument controlled by the computer, CPU and the monitor, keyboard and mouse and everything. And this is the main hardware component of the instrument. So even though they all looks integrated to each other, but basically they are not integrated. They are separate only. So this is the sample disk where we can keep all the patient samples and all. This is the reaction disk where all the reactions in the reaction cells, everything is kept in this one. And then this is the reagent disk where all the reagents are all present. So and this is the watch sample probe. So through which this is dedicated to only taking the picking up the samples. So this is the reagent probe in which all the reagents are picked up, and this is the wash station. So wash station has multiple probes, so it washes faster than our BS240 mind ray instrument. So this is the ISE unit, so ion selective electrode. So this probe is also inside, but it is protected with the steel box. And this is our ISE panel where sodium, potassium chloride and reference electrodes are all stationed. Okay, so these are all the ISC reagents. They are open because there is no need for refrigeration for this, so they are kept out of the reagent disk, because the reagent disk has got self refrigeration, so it maintains a cooler temperature, whereas these three doesn't require, so room temperature is sufficient, so they have been kept outside. Okay, so these are on another extra reagent. This is actually a washing solution, so sample cleaner solution. So the probe, whenever it requires to be washed separately, extra wash, especially after picking up a urine sample or some fluid sample and all, so the probe actually comes here and gets it cleaned up. So this is also solution that is used whenever we give sample probe wash as a maintenance. So during that time it utilizes this, whereas the second sample cleaner in COBA C311 has no use, so it is being kept only as a dummy, to make sure that the first reagent is fixed properly, right? So this instrument doesn't have a mixer probe attached to it. So in BS240 you might have seen that there is one more probe which helps in mixing R1, R2 and all. So in this instrument that is not there, because they use ultrasound waves as a method of mixing R1 and R2. So this is the area where ultrasound is there, which helps in mixing up the R1, R2 inside these reaction cells. So this is also one of the reason why no electromagnetic waves, electromagnetic interference and all should not happen near this instrument. So centrifuges, mobile phones and all should not be nearer to this instrument. So that is one important thing we need to remember. So this is the sample disk, so where all the patient samples are kept. So there are totally about 110 positions in which you can keep the samples. So they are dedicated to calibrators, quality controls, then maintenance and all like that. They have been divided. Okay, so this is the reaction cells where all the R1, R2 and samples are mixed, and through which the reading happens, and this is where the halogen bulb is present through which the reading happens. So apart from that, down if we come, this is the NaOH solution, which is used for cleaning the reaction cells and for other purposes and all. And second one, this is the acid wash. So this is also a can similar to this only, but because the amount of solution used is very less, so I have made my own can and kept it to prevent the deterioration due to air exposure and all. So this is the distilled water tank. So basically this instrument is connected to a bigger distilled water tank. So this is only inside the instrument maintenance tank, which is directly connected to the outside tank itself. So the instrument only maintains the amount of water inside that. So it has got a physical valve through which the it intakes water and consumes and all. So we need to know some of the important positions of these probes, because many of the times when the probe gets struck or some probe gets into trouble, and you need to inform me because of the red alarm that it generates, and the instrument will totally stop. So during that time I need to know exactly what is the issue. So for that purpose, I have made this extra this thing. So you might have seen that this is the sample probe, and sample probe here it has got three places. So this is the first arrow is the first place where the water keeps coming. So whenever the probe needs to wash itself, the water will come out of this. So every time when the probe comes here, the water should come out from this. If it is not coming out, then there is a huge problem present. So we need to be very careful about that. This is the place where it goes down and washes itself with the water that is coming from this area. Okay, and this is the area where it goes down to dry itself. So after cleaning with the water, it has also got a drying unit, and this is very important because when it picks up HbA1c, where whole blood only it is going to pick up, during that time it dips a lot inside this dryer. So during that time, if the probe touches any walls in this, immediately the instrument is going to stop. So we need to be careful about that part. So this is the home position. So normally when it is not working, this probe is always present in this area. So this is where water is coming for draining, for cleaning the probe, and this is where it is going to get dried. Now apart from that, when it is in movement, so it will come to these areas. So first and the foremost thing, it is the home position. This is where it is getting itself dried, and this is the first sample position. So all the outer places where the sample is kept, it picks up from this area, and then this is the second position for sample, and then third position is where whenever it needs to do an extra cleaning of the probe, during that time it comes here, and then it will, this is the place where the it will put the sample. This is the reaction disc place. So these are all the positions where this sample probe will keep moving. So we need to be careful when the instrument is in operation. So these are all the places where the probe will keep moving. So where it is struck or where is the problem, we need to know as per that during that time. So during that time, you should able to inform me exactly where the probe is struck. Okay, now this is the reaction probe, and the reaction probe has got these many positions where it will keep moving. So first and the foremost, this is a reaction cell where it will add the R1 and R2. So this is the reaction cell, and this reaction cells keeps rotating every now and then and all. So this is the position where it will add the EcoD. EcoD is a solution which it maintains the water bath. So I think I missed that point. So basically this instrument's temperature is maintained by water bath, not by any incubation or air incubation or anything. So it has got, the reaction cells are sitting inside the water bath. So this water bath needs to be disinfected every 24 hours. So for that reason, there is a solution called EcoD. So the this reaction probe, reagent probe will pick up that, and it will add it to this area. So it directly goes to the water bath so that the water remains free of any dust and other things. So this is the, so this position is only required when it is injecting the, putting the EcoD solution inside it. So this is the reaction cell where R1 R2 is getting added. This is the EcoD adding position, and this is its normal home positions where water, through water it gets cleaned, and then it comes here and gets dried. So this reagent probe also has a drying position. Now this is a position where it picks up the apparatus to make holes in the reagent. So you don't have to make holes in the cobas reagent. So the instrument automatically does it. So this is the ISC diluent, this is the ISC reference, they are the ISC solutions. And apart from that, there are various positions for the R1 and R2. So R1, R2 and some reagents. So based on that, this reaction disk keeps rotating. Based on that, this probe will come and pick it up. So these are all the positions at which the reagent probe keeps moving, and if any issue or any struck or it is got any problem, so we need to know exactly where is the problem so that it will be helpful. Okay, so this is, so this instrument, as I told you, so this instrument we never switch it off, or there is no need to switch on, because 24/365 I keep this instrument on only. So because of that reason, I am not teaching you how to switch on or switch off. So whenever the instrument is in ready position, this is how it looks like. So it has got, so there are top lines, middle lines, so like that. So this is the workstation position. So when we look at it, I will start from one end. So you can start at anywhere, but I will start at one end. So as you can see, this is, there are three squares in this part. So the first square indicates that the instrument is attached to the host computer. That means it is attached to an interface, so that whatever reports are coming, it is directly interfaced to our hospital information system. So it indicates that part. Now after this is the auxiliary units. Now this color keeps changing depending upon the situation. There are various colors for this, but important thing what we need to remember is it should be green or orange, or it may be purple. These are the three colors where there will not any problem with the instrument and instrument keeps working. If it becomes a red color, that means some major issue has happened, so the instrument will automatically go and stop. If it is black, means computer system of the instrument is on, whereas the hardware component is switched off. So because of that reason it will be black. So each color represents something, so we have to be careful about that part. Now once the instrument is started, means it from standby it starts, so there will be a sample stop timer which comes here. That means once that starts, for the next hours, like this whatever time it is showing, till that time instrument will be functional only, it will not come back to standby. So if there is no work, we have to physically give stop and make the instrument stop, otherwise the instrument will keep working only. Okay, so that is what is represented by sample stop. So during our routine classes in the lab, you can learn more about this sample stop. Then this is the username in which this instrument is run. So normally we have not given any specific username and password to any of the technicians, so it is working with the main username password only, so we'll not bother. This is the date and time at which the instrument is working, so this is also very important. Now apart from that, we have a stop. So if you click on this like BS240, everything it is doing, it will immediately stop. Plus please do remember that BS240, when you apply the stop button, immediately everything will stop, but this doesn't happen with this instrument. So this instrument will come to stop after at least 30 to 40 seconds. So it won't come to stop immediately. Whatever the movement of the reagent probe and sample probe and all is happening, that is all comes to a particular position. After that only it will going to take stop. It is not like, the wash station is down in the disk and suddenly it stops, like that it doesn't happen. Okay, now this is the shutdown. So it will work as a log off from the username also, as well as totally shutting down the instrument also. So sampling stop you don't have to learn now, only these are all the alarm. So there are yellow alarms, red alarms, like that. Any red alarm you need to call me at any time of the day and inform me about that. So whether it is night, day, anytime, wherever I am, so any red alarms you need to inform me. So that means instrument is either totally stop or it will stop in next few minutes. So media eject, again you don't have to worry. Print, it will tell all the calibration values, all the values of the test that is running and all. So pause is again to whatever it is giving, it is pause, so we don't normally use. And the start button is anything when it needs to be started, either the sampling has to be started or calibration has to be started or QC has to be, whatever needs to be started, for that it we use. Now this is how the whole thing goes. So this standby is the indication at which the instrument is, like now it is in standby. It may be in the operation, it may be in the sample scan, it may be in the pipe, it may be in the maintenance. So what is the status of that instrument, that it will keep telling. So this is very important, so we need to be alert, always alert about this part. So where exactly it is present. So this is a small introduction to the about this instrument. So last video was very long, so for that reason I have made it short. Hope you were able to understand. Thank you.